Hello, welcome to my channel. In order for you to see this tree behind me grow in height and size, in order for you and I to increase in size and height from childhood, in order for you to see all the animals around increase in height and size since infancy, there must be something behind that. Today we are going to look at DNA replication. And before DNA replication could occur, something must trigger that, and I'll share that with you. So follow me through to the end of this video, and I promise to make everything simple for you to understand. Before DNA replication would occur, or before you would see things grow and increase in size, there must be something behind it, and this is called cell division. So the cells need to divide to increase in numbers so that it could increase you in height and in body mass. So there are two ways that these cells do the work. The first one is mitosis, and mitosis is called copy division. That one, it copies the cell ditto. So for instance, if you have a cell containing a chromosome number of 46, just copies exactly the same thing. Yes, that is the first one. And the second one is meiosis or meiosis. Anyway that you mention it is allowed. And that one is called reduction division. So meaning that one, it reduces the chromosome number. So for instance, if it is 46, then it will reduce it to 23, half. Okay. And this happens so that we could get diversity during selection. Okay. And it will happen, this also happens for sexual reproduction to take place. So for instance, the male will bring a chromosome number of 23, the female to bring a chromosome number of 23, and in all it becomes 146. Okay. So but today we are not on cell division. So by popular request, if you want me to do a video on cell division, on mitosis and meiosis, I would definitely do that. But today we are going to look at DNA replication, and I'll take you through all the 5 steps that DNA passes through to make a copy of itself. So DNA replication simply means DNA copying itself, okay, or DNA multiplying itself, increasing itself. And this happens because the cell is dividing. So this is when the cell begins to divide, because the cell, as it's dividing, for instance, the meiosis, the cell needs to divide into 2. So it has to copy the DNA, the identical DNA, into the other cells that is dividing. So the same DNA will be in the other half of the cell. So this is where the replication need to occur. So now let's get into the DNA replication. So for DNA to begin to replicate, and before that, just see DNA to be this, okay? It's double, right? A double-stranded DNA, okay? The DNA is double-stranded. So take it to be like this, but it is helical in shape, leather-like, but it is twisted around, okay, but not as straight as this. But because I want to explain something for you to get, I want you to take it that this is your DNA, okay? It is double-stranded. All right. So the first stage in replication is unwinding the double strand, okay? So the double strand, if we say unwind, meaning opening up, the double strand needs to open so that we can make a copy of this and make a copy of this, okay? So this means that we would need somebody or an agent, or let's say an enzyme, to do this work, and this enzyme is called helicase. So the enzyme that is responsible for the unwinding of the DNA is called helicase. Okay. So after the DNA has been unwound. You go to 2. Step 2, we will need an enzyme to stabilize it. The reason, what do you call it? Sorry, DNA is double stranded is if it becomes single, it becomes unstable and we can easily lose it. Okay, so it needs to be double stranded. So because we are going to do the synthesis in a very short period of time, we would need a body that will be able to sustain it or stabilize it through that replication process. Okay, and these are protein, single stranded protein. Okay, so single stranded proteins help to stabilize the unwound DNA, the single stranded DNA. Okay, so after it has been stabilized by these proteins, then DNA begins to synthesize continuously from 5 prime to the 3 prime end. So DNA, as you see it, is being labeled 5 prime to 3 prime. So for the one on top, so 5 prime at the tip here to 3 prime at the end here, and the opposite one will be 3 prime to 5 prime. So they are in opposite direction, they are moving in opposite direction. So, and they say DNA begins its synthesis from the 5 prime to 3 prime. So the first one 5 prime to 3 prime, the second one 3 prime to 5 prime. So let's say that synthesis is supposed to begin, then the 5 prime will begin at the 3 prime end of the old one. Okay, so the synthesis will begin from here and it will be continuous, and this synthesis will be made by the enzyme DNA polymerase. So it is the DNA polymerase that would elongate, okay, synthesize or produce the other strand on the old one, so it produce a new strand on the old one. Okay, so keep that in mind. So we have 5 prime to 3 prime. Okay, so after the DNA synthesis starts and continues from the 5 prime to the 3 prime, and we move to step 4. Step 4 is where primase, RNA primase. Okay, so these are primers, RNA primers that initiates, and a primer is a short piece of RNA that initiates the DNA synthesis. Okay, that will initiate the DNA synthesis. So the primer would initiate the DNA synthesis, and it will later be replaced by the polymerase, DNA polymerase, and it will elongate or continue the synthesis. But on the opposite strand, that one is not a continuous, it's a discontinuous synthesis, meaning it does it in breakages, in batches. So if it begins the synthesis from here, stops, jumps, makes a gap, then goes to the next one. And these fragments are called Okazaki fragments. Okazaki fragments. Okay, so at step what? Which step is that? Step 4. Yes, at step 4, we have a discontinuous synthesis. Okay, and this discontinuous synthesis is started by an RNA primase or a primase, which is an RNA primer initiates the synthesis, then the DNA polymerase takes over. And it doesn't do it in a continuous form, it does it in a broken form or discontinuous form. And that is where we would get our fragments called Okazaki fragments. And the fifth one is where these fragments, Okazaki fragments, are being joined together by DNA ligase. Okay, so they are being joined together. So it makes now the DNA synthesis complete by joining or fixing the gaps. Okay, so this is done by DNA ligase. So this is how DNA replication occurs in living cells, okay, or inside a living organism. Let me take it all over again. DNA synthesis begins with the unwinding of the double helix or the double-stranded DNA by the enzyme helicase. And after it has been unwound, we need to stabilize the single strand. So that's where the enzyme or proteins, single-stranded proteins, stabilizes the strand. And after the strand has been stabilized, then synthesis begins continuous from 5 prime to 3 prime. And as it's going on, we also have in the opposite strand primase. Okay, that's an RNA primer, initiates the synthesis, then the DNA polymerase takes over and makes short synthesis. That's called the Okazaki fragments. And these Okazaki fragments are being joined together by DNA ligase. And this is the whole replication process that occurs in a living organism. Stay tuned. Thank you for watching or making it up to the end of this video. Next time, we would be looking at DNA replication outside the living organism, which is called the Sanger sequencing. Don't miss it. Always come back here if you like more of these videos. Share to your friends to also learn something new. See you next time.